Welcome back to Cell and Molecular Biology course. I'm your course instructor Inia Tendril. In this video, we will explore the fluid mosaic model of the plasma membrane. We will break down the structure of the plasma membrane, exploring its various components and how they function together to maintain cellular integrity and regulate the movement of substances in and out of the cells. By the end of this video, you will have a clear understanding of the dynamic fluid nature of the membrane and its critical role in cellular biology. Now let's begin with the introduction to plasma membrane. The plasma membrane is a thin, flexible barrier that surrounds all cells, providing a boundary between the internal contents of the cell and the external environment. It plays a key role in regulating what enters and exits the cell, ensuring the proper environment for cellular processes to take place. This barrier, though only a few nanometers thick, is crucial for maintaining homeostasis. Now let's see what is the fluid mosaic model. The fluid mosaic model was proposed by Singer and Nicholson in 1972, which describes the plasma membrane as a mosaic of diverse protein molecules embedded in or attached to a fluid lipid bilayer. The fluid nature of the membrane is due to the ability of lipids and proteins to move sideways within the layer. This provides flexibility, allowing for various interactions and functions. In this illustration, you can see the phospholipid bilayer, and then you can see the peripheral and integral proteins, and glycoproteins and glycolipids on the external side. At the heart of the plasma membrane is the phospholipid bilayer. Each phospholipid molecule has a hydrophilic or water attracting head and hydrophobic or water repelling tails. You can see in the illustration the phospholipid structure. You can see a head part which is in purple color. This is the head part which is hydrophilic, and the hydrophobic part is facing inwards. You can see a straight line which is the saturated lipid tail, and the other one is which is not straight is the unsaturated fatty acid. You can see the head part which is hydrophilic and the tail which is hydrophobic. You can see a straight line which represents the saturated fatty acid, and the other one which is not straight, you can see a bend. It represents the unsaturated fatty acid. So this is the structure of the phospholipid. In an aqueous environment, these molecules arrange themselves into a bilayer with the hydrophobic tails facing inward and the hydrophilic heads facing outward, creating a stable barrier between the cell and its surroundings. In the illustration, you can see the phospholipid heads facing outwards and the hydrophobic tails facing inward. So this is a phospholipid bilayer structure. Now let's see the proteins in the membrane. Proteins are an integral part of the plasma membrane. They come in two main types: integral proteins, which are embedded within the lipid bilayer, and peripheral proteins, which are attached to the surface. These proteins perform a variety of functions, including transporting molecules across the membrane, serving as receptors for signaling molecules, and they also provide structural support. Now let's see cholesterol in the membrane. Cholesterol molecules found within the phospholipid bilayer play a key role in maintaining membrane fluidity. They prevent the membrane from becoming too rigid in cold temperatures or too fluid in warm temperatures. Thus, they ensure that the membrane remains stable yet flexible under varying conditions. In the illustration, you can see the cholesterol molecules. It is also mentioned that the cholesterol asymmetry, that is, in the both the external and internal layer of the plasma membrane, you can see the cholesterol interspersed within the layer, and it is not uh, equally present within the cell membrane. Glycoproteins and glycolipids are important for cell-cell recognition and communication. These molecules they consist of carbohydrates attached to proteins or lipids, and they project from the outer surface of the membrane. They act as identification markers, allowing cells to recognize and interact with each other, which is vital for immune response and tissue formation. In the illustration, you can see glycoprotein, glycolipid, and proteoglycan. These glycoproteins and glycolipids, they also help in self-recognition. That is, uh, usually immune cells respond to non-self molecules. And how would they recognize that if the cell is self or non-self? It is through these structures, that is glycoproteins. One of the key features of the fluid mosaic model is the fluidity of the membrane. The lipid molecules and some proteins are able to move laterally within the layer, giving the membrane a flexible structure. This fluidity allows the cell to adapt to changing conditions, and it enables them to repair itself and facilitate the movement of materials. The plasma membrane serves several crucial functions. It controls the movement of substances in and out of the cell, allowing essential nutrients to enter while keeping harmful substances out. It also protects the cell by providing a stable boundary. In addition to that, the membrane plays a role in cell signaling, allowing cells to communicate with each other through receptor proteins. and chemical signals. In conclusion, the fluid mosaic model provides a comprehensive framework for understanding the plasma membrane. It's structured, a dynamic combination of lipids, proteins, and carbohydrates enables it to perform essential functions that are critical to the survival and operation of the cell. Uh, Thus, by maintaining fluidity and flexibility, the membrane adapts to environmental changes and it ensures that the cell can function efficiently in response to various stimuli. This model remains one of the foundational concepts in cell biology, shaping our understanding of cellular structure and behavior. Thank you.